Don't poke anyone's eye out, okay? Thanks, guys. Thanks, Angela, for a nice introduction. I'm here to talk about a genomics consortium that we've been working on. It's a little strange to give a talk about a genomics consortium at a genomics consortium meeting, but it's okay. Before starting, I just want to give a quick shout out to Tara Eisner. She's my co-founder of this effort, and there's no way it could have been started or gone as far as it did without her. She was supposed to be here today, but she just couldn't make it, so she feels bad she couldn't be here, but just, keep her in mind. These are my financial disclosures for my burgeoning financial empire. I think that's all right. We can skip that. Okay. I think I'll just start by just saying that molecular pathology has gotten really quite a bit more difficult and challenging over the last number of years, I think, as we all kind of know. When I was doing my residency training and even, in fellowship, if you wanted to look at a target, you did a little PCR. You could analyze it by running it on a gel or Sanger sequencing. It was pretty straightforward. It seemed complicated at the time, but I think looking back on it, it wasn't so bad, and the interpretation was fairly straightforward. And now, looking forward even 10 years from that point, right, we've got these highly complicated next-gen sequencing platforms. The library preparations that we have to do to get ready for the sequencing are really complicated. And then the sequencers are producing all sorts of big data, and it forces you to think about, what kind of large-scale computing platforms you're going to use, and hiring bioinformaticians and making pipelines, and then you have to think about how to interpret all of the information that you're getting, and then how to integrate it all in your lab information system. So it really is, it's been a fun challenge over the years to bring this into the lab, but it really is tricky. And one of the things I think that really held us back as a community was that all of us in the academic space, we all sort of worked on this independently. So we were kind of in a siloed type fashion. We were doing all of our validation, and so was everybody else in all of our development. And everyone was, trying to solve the same problems in parallel, all the different difficulties with variant calling. Everybody was working on them with their bioinformaticians in silos and not sharing. And we were kind of competing with each other in ways that, in retrospect, don't really make any sense, because we don't really actually compete with each other. We're really kind of in the same community. Just looking, just to give a little overview, this, in 2013 I was brought on and we started putting this panel together, 2014 to 2015. And, what we wanted to do was bring on a 1,000+ gene panel, not to report all the genes because we don't even know what to say about them. So we report a, subset of the genes. And then also have a lot of different features in there, like copy number calling and microsatellite instability, viruses, fusions, et cetera. And so when you're trying to put together a panel like that, especially of that size, you're really kind of, your real main option is really to do hybrid capture sequencing in order to do it. And I'm trying to go a little bit quickly. I know there's a boat ride that leaves in a little bit, so I'm just going to try to get through, but catch up a little bit. But at any rate, if you're not familiar with hybrid capture sequencing, although I bet most people in here are, I'll just go over it just very quickly. In general, you have your genomic DNA. And the first thing you're going to do is, fragment it into small fragments and make a library, and that's sort of a whole genome sequencing library. And imagine that there are particular fragments in there that you want to sequence. Those would be those dark blue fragments. And then there are many fragments that you don't want to sequence. Other genes that you don't care about, junk DNA, et cetera. So what you want to do is set up a system to purify the dark blue fragments away from the light blue fragments. And the way that that's done is to create a panel of oligonucleotide probes. These are just individual oligonucleotides. Oops, I need the back button. Oh, geez, the laser. Sorry, guys. So you manufacture a bunch of oligonucleotides that are biotinylated, and they have the sequences that match the dark blue fragments, and then you can set up a hybridization reaction, and if your oligos are there and your fragments are there, you will have a successful hybridization, and you can pull them down with magnetic beads, and then you've got an enriched library to sequence. So the key component here is really this panel of probes that you're putting together. It really, highly dependent on that. And at the time that we were starting this, there was really only a couple of options for making that panel of probes, and that was to work with one or two different companies. And you would basically have to say, okay, what are all the regions of the genes that I want to capture, the different exons of the different genes, and you would know their genomic coordinates, and you'd make a big list, and you'd send it to the company, and they would say, okay, well, then you need to make these particular probes in these sequences, and they had a machine that would produce the probes all in one big batch. It had a lot of different spots, and it would program a different spot on the machine for each different oligonucleotide, and then you would push go, and all of your probes would be created at the same time on one machine, according to the design. And that generally actually worked quite well for most genes, and we didn't have a problem with it. But for certain genes, it was really an issue. And the difficult spots for the most part are areas where the genomic sequence has a lot of GC-rich content. So if the bases are largely Gs and Cs, it can cause a lot of secondary structure and problems, and you would have some serious dropout in some pretty bad areas. So this was CEBP alpha. So we wanted to do heme malignancy, but how do you do heme malignancy tumor profiling if you can't get CEBP alpha to work, which is one that's needed even for diagnostic criteria in AML. And you can see here the depth of sequencing we're getting is good over here in the UTR that we don't care about. This is the coding area, and here there's very little sequencing data at all. You can't call any mutations. It's a real mess. We tried resynthesizing, boosting probes, all that stuff. We spent a bunch of money doing that. Didn't work at all, and we got to a point where we thought we were really going to have to either cancel the project or downsize it or really rethink what we were doing. And those were not good days until just fortunately we were just meeting with our rep from IDT, which is a company that makes oligonucleotides for a long time, and they were getting into the next-gen sequencing space, making probes. And they said, oh, this is easy. This is not a real problem. Just buy some of our probes, spike them into your panel. It's going to fix it. And we had become conditioned to think that high GC DNA was just not really easily approachable. So that we had nothing to lose, so we bought some probes and we spiked them in. And you can see we went from a profile of this with no data to a wealth of sequencing data, overnight with the addition of a couple of $20 probes, and that really changed our mind about what it means to do sequencing and how things work. And we started getting greedy and using that technique everywhere. We were having problems, and it really worked. The reason why this works, we had come to think that high GC DNA is just difficult to capture, but it's not. What it is it's difficult to manufacture high GC probes. And when you have this bulk synthesizer, it just doesn't make your probes. They just weren't able to do it. Whereas IDT is making them one at a time, doing quality control. If it's not right, they make it again. And so you know that you have the probes that you were going to buy. So at that point, we were like, oh, well, if that's really the way it is, why don't we just go ahead and buy everything from these guys and just reconstitute our panel with stuff from IDT? Because we had been paying our original vendor like $40,000 every time we were going to do 1,000 samples. And we thought, okay, well, maybe if we just buy them as a large bulk, like a large reference laboratory, maybe it'll be better. And so we asked them, and they said, okay, well, you need to make about 50,000 probes. It'll cost you about $300,000 to $400,000. And at that point, we were like, oh boy, that's kind of expensive. We didn't know 100% if it was going to work or if there were issues with it. And so we kind of shelved the idea a little bit until we asked them, wait a second, if we do this, like how much are we actually buying? And the truth is, you're buying individual probes, so many of them, and then mixing them all together, then you end up with a boatload of probe, which turned out to be good for 30 million samples. So at that point, like to put it in context, we do 2,000 samples a year, so that's 15,000 years worth of stuff for the lab, So, it's a lot, and it didn't take a lot of brain power at that point to think that maybe this is something that could be shared with another lab, right? There's a total wealth of stuff, and we could maybe lower the cost by sharing. So that's when I started talking to Dara, and we thought maybe we'll try to share it, the two of us, and still it's like $150,000 to $200,000. It's a little pricey. But then we thought, why don't we try to get some more labs and see if we can get like a handful of labs and make it reasonable. And so that's what we did. We started talking to all of our friends at different labs, and we set up WebExes, and we told them this is our idea and this is what we're going to do. And we were just hoping to get like 3 people to join us who were willing to spend some money on a possibly crazy idea. Fortunately or unfortunately, we ended up with 15 labs that wanted to join, and now we've got 17 laboratories that want to participate in this purchase together. And that made it a lot, higher risk, a little daunting, and also what are we going to buy? Because our original plan had just been to buy my panel and share it, but these guys all had their own panels. The gene list requests at the different places were anywhere from 100 genes to 1,400 genes. There was no way we could agree, and more than half of the genes were requested by only one lab, and nobody else wanted them. So what are you going to do? So we decided the only way to actually make it work would be just to buy every gene that every lab requested, a total of 2,640 genes worth of probe material. And the way we decided to do it, I think, was kind of creative. We decided to have these matrix plates and to put the probes for each gene in one well, so that all your probes for KRAS are here, and EGFR are here, and TP53, so on, so that you get all these plates. And then if you want to put together a 1,000 gene panel, you pick out the genes that you want and pipette all those together, and somebody else can make a 500 gene panel that's totally different. And everyone can make their own panel that they wanted, and we don't have to dictate panels and have everybody agree on genes, because that's just really difficult amongst a group like this. So that's what we did. We ended up buying 95,000 plus probes in 2,670 pools across like 28 plates times 2. Everyone's getting 56 plates worth of material times 17 sites. Everybody paid only about $25K for this, which is essentially like many years worth of supplies for the lab. And IDT put all that together within 6 weeks and delivered it. And then we could see if it worked or not, and it was a little bit of a fingers crossed moment, and it actually ended up quite nice, right? So for most genes, we don't see really a difference. If you don't have a high GC problem, it works fine for both. But here's ARID1A exon 1, which is a heavily GC-rich gene, and you can see our prior panel had a real problem with it, and here we don't have a problem anymore. And so it just made things quite easy, including our revalidation of our panel. We made a new panel, and you can see here variant frequency head-to-head for a lot of samples, and then collectively, and then also across indels. So it was just quite straightforward to revalidate and switch over to the new probe set. And it wasn't just us. Here's the University of Washington, same thing, nice data profile. And they published this, and here's Johns Hopkins with their panel, and more and more. And so we would go and, Dara and I would give this talk. We gave this talk at AMP a couple of times. And every time we gave this talk, the people would come up to us and say, like, how can we join? Is there a way we can get in on it? And we ended up doing a secondary purchase and added another 12 laboratories a couple of years ago. So at this point, we are up to 29 different academic centers working together on this stuff and using similar chemistry in our panels. And 14 of the sites are already live with clinical panels in their lab, which are live. And so we estimate somewhere around 25,000 cancer samples a year are processed through goal panels. So that's kind of nice. One of the things we thought, when we originally did this, we thought everyone would just get the probes and then they'd say thanks and then they'd go back to working independently and that would be kind of the end of it. But that's really kind of not what happened at all. People just started asking each other questions like, what library prep are you using? How are you doing this? Can you share some samples with me? What are you doing for bioinformatics? And it really kind of turned into a working group, sort of organically, which was nice. And Dara and I, at that point, we kind of thought, we had thought the purchase itself was the end, and then we kind of realized that the purchase itself was really the beginning of the project, and there was really like a ton more stuff that we could do, beyond purchases and validation support, looking at scientific projects or, advocacy, a variety of things. So what we did is we decided to turn it into a formal nonprofit organization, which is what we did a couple of years ago. So we talked to the Association for Pathology Chairs, which is a really nice organization, and they have kind of the same mission as we do, which is just to support academic pathology. And Priscilla Markwood, who's their executive director, has been kind of like an advice guru for us, and Mel Limson is their director of programs and development. He's been working for us part-time, and he's just done an amazing amount of work. We put together a board of managers, Jen Morissette from Penn and Chris Goki from Hopkins and Dan Jones from Ohio State. So we have a board. It's a real organization. We got our 501c3 status approved, and we got some bylaws, et cetera. So we're really just getting started. So I just wanted to take a minute to talk about some of the stuff that we're doing collectively as a group. The first thing that's of interest to us is trying to do an actual concordance study between our labs. And the reason for that is really because there's a lot of speculation and doubt out there about how our lab developed tests are and how do they compare with each other and what's the quality. And we thought it would be nice to show how we're doing. And also because all of our labs are using similar chemistry, we thought it would be a good opportunity to actually show some high quality data. So what we did is we crowdsourced a whole lot of DNA samples from the labs and tested them. We got 9 labs with approved panels to go in on it. And the idea was that each one would run it on their own, get their raw data, process it on their own informatics, but also we set up a cloud-based consensus pipeline. We actually used the Illumina Dragon semantic pipeline, which is a really nice off-the-shelf pipeline from Illumina. And everyone could take their raw data and we could process it through that and get a different set of results. And then we could start to see, if there was some discrepancy in the original results, but it gets fixed by having a communal pipeline, then you can tell that it's actually a bioinformatics issue versus a wet lab issue, and we can start digging down into like what contributes to discordance. So this is the raw results of the individual site pipeline analysis. It's a little tricky to say, but these are 100 different variants in 25 samples that were tested. This, for example, is one variant that was identified by all labs at around 70% VAF. And so you can see we're generally quite concordant between the labs. There is some discrepancy here, but the overall sensitivity is still 98.3%, which is pretty reasonable. And if you look at what's discordant, it's really mostly homopolymer length artifacts. Here you can see a large homopolymer site next to this exon. We got clean sequencing data over here, messy sequencing over here. That's very characteristic of Illumina sequencing. Once you get one of these homopolymers, the PCR can make length affecting mistakes, and so can the sequencer, and you end up with a collection of different size errors around them. And it can be hard to tell if there's a real mutation there or not. And it's a problem for many labs. Different labs are going to have different filters set up. Even if you look at Foundation Medicine, they'll say these are sources of reduced sensitivity. And we had a lot of these in our sample set because we picked a bunch of microsatellite unstable cases. This is a microsatellite in order to have a lot of mutations that we were going to look for. So a little overestimate of the impact of this on the sample set, but also to be expected because every lab is going to call these a little bit differently. So if we take the individual pipeline results and now we pass it through the communal pipeline, the amount of discordance has been markedly reduced. The sensitivity goes up to 99.7%, which we thought was really nice. And then you could ask, okay, well, you guys agree with each other pretty good, but are you making mistakes or are you correct? So we've endeavored to go after that by doing some digital droplet PCR on some of these variants, and we've picked 13 variants. And the digital droplet result is this rectangle, and you can see that the lab results all cluster nicely around that. So we're actually doing, not only are we agreeing with each other, but we're actually like kind of on target. So I think overall, it really, it's kind of nice. It sort of supports our hypothesis that shared probe chemistry can facilitate uniformity and concordance between labs. Of 898 expected variant calls, only really one was discordant due to a wet lab issue that we identified. And most of the discordance that we see comes at the bioinformatics level, and most of that can be fixed or at least harmonized by using a communal pipeline. So there's a lot of important QI feedback to the labs that came from this, and now we can start looking at our homopolymer length calling and seeing what's the ideal way to do this as a group. Now this is just a preliminary analysis. Future stuff is we're going to look at copy number variants and TMB and all sorts of other things out of this data set, but we're still working on that. So, who knows, stay tuned. The concordance study isn't just to show how we're doing. The point really is to use the concordance study to help the labs in a variety of ways. One of them is to take it and say, hey, we're doing a good job. There's lots of people who say that we're the Wild West of medicine, and I really can't stand that. Like, we're not cowboys. Like, we're actually trying to do a good job. It's not, I don't know. It's not the OK Corral at any rate. So, we can take the results of this and share it with the stakeholders, regulatory stakeholders, and payers. We might be able to convince pharma companies to treat us as a site to do a multi-site clinical trial. And then we've saved extra DNA for every one of these samples for labs that join in the future. They can get samples of these highly vetted samples to help with their validations. The next thing we're looking at is actually getting into data sharing. As you guys probably are all aware, like we have our labs, but at least on the germline side, you have good resources. We have some emerging resources for looking at rare, weird variants. But all the time, I sequence a sample and I want to know, hey, I've never seen this before. Has anybody in our group seen it? Has Dara seen it? Has anyone seen it? And it's hard to look up that information, and not only to look it up, but to see how somebody graded it and what did they actually say about it. And so we're looking into creating a shared database that we can all deposit all of our data into on a case basis. And so then you could see, look it up and see, okay, has anyone seen this variant before? What type of tumors? Where was it graded? And you know it's vetted because it's all being done by sequencing among the group with similar chemistry, et cetera. And I think this could really help us not only reduce the amount of interpretation time that's needed, but actually help us start to move towards harmonizing our reporting between labs, because that's something that seems to be, to have some differences between labs, not just grading scales, but also how we view these variants between our labs. All right, in order to do that, we really needed to get some help getting started. We thought we were actually well positioned to facilitate a variety of types of trials as a network. Dealing with pharma companies, like a pharma company could come and ask us then, based on having this data shared, can we do data mining analyses, or can we even retest interesting specimens or do ring studies? There's a lot of possibilities for linking our group to various different parties. And so we started out after going to APC by going and trying to raise founding funds, and went to different pharma companies and said, hey, can you help us get started? And we were really fortunate. We were able to raise over $1 million from AstraZeneca, Genentech, Loxo, Amgen, and Blueprint Medicines to get us started. And we've been using those funds to hire project managers and do a lot of the other activities that we've been doing as a group. And so thanks to our sponsors. I always like to say that, and I really appreciate it. In order to, as a first step, if you have a pharma company and they want to come to a network of academic centers and start a project, if there's 29 academic centers and they want to start a project with us, without any kind of other support, they really kind of need to make a legal agreement with 29 different academic centers in order to get started. And if you've ever done a legal agreement at an academic medical center, it's terrible. So trying to do that is challenging, and it's just challenging. So what we wanted to do is get set up so that we actually had the legal framework in order to do this, by setting up a hub and spoke model whereby the goal independent organization is the hub, and we can make a master agreement between us and all of the different sites that covers things like data sharing, projects, MTAs, intellectual property, et cetera, so that we could be a one-stop shop and a funding source from a pharma company. They could come and say, hey, we'll sponsor this project and we can publish the results, and then they can come and make just an agreement with goal, and all the other legal framework is already handled. And so that's our sort of main goal for this year. We've got 7 of these contracts fully executed and a bunch of the other ones in process. And if we can finish that this year, it'll be really nice. So that's probably our most important thing to finish. I'll just stop there. I think, I hope you can see we're trying our best to bring value to the community and try to raise all the boats in our network. I won't go into the details on each of these things, but we really are trying to help everyone. And I think we've got engagement on that basis, because people seem sort of to really gravitate to it because it seems to be helping everyone, and that's just a nice feeling. So I'll just stop there. We are always interested in accepting new member sites if anyone wants to join. We're working on another purchase now for another group of sites. If anyone has a lab that might benefit in any way, just you can always talk to me. My contact info will be on the next slide. Just want to thank everybody involved. These are people, not even a full list of people, but people at all of the 29 different centers. Also wanted to thank one person who's here who works at QIAGEN now, Chowji, who he used to work in our lab, and he was really instrumental in getting a lot of our stuff set up. So thank you. And that's about it. My info is here. If you want to look at our website, you can click the little link. And I don't know if we have time for Q&A, but we'll see. But thank you very much.
Thank you so much, Jeremy. I know some people do need to leave for their activities, and that's totally fine. This is such a great initiative. I think for those that do want to stay, I think we can take maybe 1 or 2 questions. But you could always find Jeremy later as well with his contact info.
Hey, Jeremy, Shrey here. Nice to see you. And fantastic talk. Using Illumina Dragon to process the bioinformatics data, have you faced, so I know that with the labs at Chicago and UPenn, we used to customize the bioinformatics pipelines and just solve the problem right away or present with the request at least. So how do you envision that with Illumina Dragon? And are they very convincing in those aspects? Because it's a lockdown pipeline and
Yep, it's sort of a lockdown pipeline, but I've actually been working with the team that developed it, and they're interested in trying to modify it or change certain things to help us make it as clinically valuable as we can make it. So, we were at 99.7%. There's a couple of things that weren't quite there, and especially, the 97.7% really refers all those to the main pathogenic findings in those samples. When you go beyond those, there's a couple of other little funny things that because of the way their variant caller works, you would expect to pick up but maybe don't. And they have an alignment-based system for calling, and they also have a column-based system. And so we've been exploring the column-based system, and it seems like it's a potential addition or potential alternative. So their whole team has been working with, I guess, me mostly on trying to go through these samples and trying to figure out if there's a better way to call it. And then we can, turn it into the modified goal pipeline as we go. And that's really the hope, is to get it to be as good as we can make it as a group, not just use the off-the-shelf platform. So they've been really helpful with that.
Yeah, I mean, with the sensitivity, there's no question Dragon is performing really well. My question was more towards adding new modules, like calling variant, like MSIs, TMBs, and other things down the line, HRD and things like that you may want to implement. And I think Illumina is working in that direction. But then how much? So you said that
it's all doable. You do it through the Illumina Connected Analytics platform, which I'm not an expert at using that platform. They've kind of been helping me, but you can take the Dragon pipeline and you can add modules to it in that system. So theoretically you can add kind of whatever you want to it.
Right, the graphical version of that.
Yeah, or even the command line version. There's multiple different ways to do it. Yeah.
All right, sounds good. Thank you.
Sure.
All right, thank you, Jeremy. I think we will end for today, so everyone can relax now, but you can find Jeremy afterwards. He'll probably stick around for a little bit, and I'm sure he'd be happy to take your questions. Thank you, Jeremy.